How do T cells use MHC in the immune system? Have you ever wondered how our body identifies and fights off infections? One key player in this process is T cells, which relying on Major Histocompatibility Complex molecules, often referred to as MHC. These molecules are essential for T cells to recognize threats in our body. Let's break down how this works. MHC molecules are proteins located on the surface of cells. They display small fragments of proteins, called peptides, from inside the cell or from invading pathogens. This presentation is like a warning signal, alerting T cells to potential dangers. There are two main types of MHC molecules that play distinct roles in T cell function. The first type is Class I MHC molecules. These are found on nearly all nucleated cells in the body. They present peptides that come from proteins made inside the cell. For example, if a cell is infected by a virus, it will display viral proteins on its Class I MHC molecules. Cytotoxic T cells, also known as CD8-positive T cells, recognize these Class I MHC peptide complexes. When they detect a foreign peptide, they spring into action, targeting and destroying the infected or abnormal cell to stop the spread of infection. The second type is Class II MHC molecules. These are mainly present on specialized cells called antigen-presenting cells, which include dendritic cells, macrophages, and B cells. These cells take in proteins from outside, process them, and then present peptide fragments on Class II MHC molecules. Helper T cells, or CD for positive T cells, recognize these Class II MHC peptide complexes. When they do, they activate and coordinate other immune cells to mount a strong immune response. T cells use their unique T cell receptors to recognize the combination of a peptide and an MHC molecule. In addition to this receptor, T cells have co-receptors, CD8 for cytotoxic T cells and CD for helper T cells. These co-receptors bind to specific parts of the respective MHC class. This dual recognition ensures that each T cell type interacts correctly with the right MHC class and target cells. This is vital for preventing the immune system from mistakenly attacking the body's own healthy cells. When a T cell receptor binds to a peptide MHC complex, it triggers signaling inside the T cell. This leads to its activation, proliferation, and differentiation into effector cells. These effector cells are the ones that carry out immune functions, such as killing infected cells or helping other immune cells respond effectively. In summary, the interaction between T cells and MHC molecules is a fundamental mechanism in our immune system. It allows T cells to monitor cellular health and coordinate targeted immune responses. This process is essential for our body to detect and eliminate infected or abnormal cells while ensuring that it does not attack its own tissues. Understanding this relationship helps us appreciate how our immune system works to keep us healthy.